Good evening, everybody. We are going to start the session shortly. All right, everyone, let's start with today's session. So in the last session, we had done till this question. Okay, all right. Now the next question, XO type of sex determination and XY type of sex determination, they are the examples of. So in today's session, we'll be taking up the questions mainly on sex determination part. All right. Find out the answer. XO type of sex determination and XY type of sex determination, they are the examples of. Very good, Kashish. Very good, Ira. Okay, Mahek. Go through the questions again. Go through the options again. Darshini, put your answer in the chat box. Yes, Mahek. Now it's correct. All right. Okay, so now see, let's understand what the question is saying. The question is asking that XO type of sex determination and XY type of sex determination, they are the examples of. So now see, when we are talking about XO type of sex determination, in this case, if I talk about XX and XO type of sex determination, here a female is autosomes plus XX, right? 2A is representing the autosomes plus XX, and male is 2A, again 2A is autosomes plus X. So now if we see the type of the gametes that are going to be formed here. So for the female, it is going to be A plus X, right? Here A plus O, that means no X chromosome, and A plus X, right? So here we can clearly see that here we are getting, in case of males, we are getting two different types of gametes. So male is heterogametic in this case. Right, now let's understand the next type, that is the XY type. So again here XX and XY type. Here a female can be represented as 2A plus XX, right? And if we talk about the male, then 2A plus XY. If we see the types of the gametes that are going to be formed, here A plus X, A plus X for the females. In case of males, it is going to be A plus X and A plus Y. And again, I'm repeating this part. A is representing the autosomes. All right. So again, we can clearly see here what is happening. In case of males, we are finding two different types of gametes. Heterogametes. Right? Male is heterogametic. What will be the answer? A. Male heterogameity. All right. Moving on to the next question. Grasshopper is an example of XO type of sex determination in which the males have. Yes, everybody put the answer in the chat box. This is an easy one. Right, Darshini, Kashish, correct answer. Good. Mahek, correct answer. Yes, Ira, correct answer. Right? In grasshopper, what are we going to find? We are going to find again XX and XO type of sex determination. Here, if I want to talk about the genotype of female, it is going to be 2A plus XX. However, in case of males, it is 2A plus XO. That is only one X chromosome is present. The males, they have one X chromosome. Okay. Next, in XO type of sex determination. All right, again, a related question. In XO type of sex determination, what is happening? Correct, Mahek, Darshini, Kashish, Ira, correct answer. Good. Right, right. We've already seen in case of XO type of sex determination, male is, the genotype of male is 2A plus XO. So the gametes that we are going to find is going to be A plus O, that is absence of X chromosome or presence of X chromosome, right? So here what are we finding? We can clearly see that the males are producing two different types of gametes. All right, so B will be the answer. Good. Next question. Select the disease which is caused by recessive autosomal gene when present in homozygous condition. Good, Krishna, correct answer. Good, Ira, Sapna, correct answer. right? Good, right? It is asking about the autosomal recessive disorder. All of the given options, they fall into the category of autosomal recessive disorder, right? See, what what is autosomal recessive disorder? Autosomal recessive disorder, you inherit two mutated genes, one from each parent, right? For a recessive disorder to be expressed, two mutated genes are required. These disorders, they are usually passed on by two carriers. Their health is rarely affected, but they have one mutated gene, that is recessive gene, and one normal gene for the condition, right? See here in the chart, there are carrier parents. The male is also a carrier, the female is also a carrier, and as a result of that, there are chances of being, or of the offspring to be affected. All right, next question. Trisomy is represented by, how do we represent trisomy? Good, Kashish, very good. Yes, Darshini, correct answer. Good. Everybody put your answers in the chat box. See, what is the meaning of trisomy? What is the meaning of trisomy? That here a person has three copies of chromosome instead of two, right? Normally, we have two copies of each chromosome, one from paternal side, one from maternal side. But in case of trisomy, what is happening? Three copies of chromosomes are happening instead of two. Okay, like one of the classic examples of trisomy is trisomy of 21st chromosome. In which disorder does this occur? Yes, put your answers in the chat box. In which disorder are we going to find trisomy of 21st chromosome? Three copies of chromosome number 21. Perfect. Very good, Darshini. Very good, Kashish, right? Trisomy of 21st chromosome we are going to find in Down's syndrome. So how are we going to represent it? We are going to represent it like this. 2n plus 1. There is one extra copy of a chromosome. Okay. Okay, see here, normally individuals, they have 23 pairs, that is 46 chromosome. In a rare genetic condition called trisomy, there are 47 chromosomes. So if the chromosomes are XX, it may be triple X or it may be XXY. And if the chromosomes are XY, it may be XYX or XYY. Okay, like trisomy of chromosome number 21 causes Down syndrome. Then there is trisomy in 13th chromosome also, which causes Patau's syndrome. All right. So these are certain examples. These are the examples of the trisomy. Next, Mongolism is a genetic disorder which is caused by presence of an extra chromosome number. What is Mongolism? Good, very good, Darshini, very good. Kashish, correct answer. All right. Yes, Mongolism, we are talking about the Down syndrome only. And we have seen in case of Down syndrome, what is happening? Trisomy of 21st chromosome. Okay, so answer will be B21. Next question. All right, see here, Down syndrome, trisomy 21 or Mongolism. It is a congenital disorder caused by presence in the human genome of extra genetic material from chromosome number 21. See here, the karyotype which is given here, we can see that there are 3 copies of chromosome number 21 present. So this is Down syndrome, or which was previously called as Mongolism. Klinefelter's syndrome is characterized by a karyotype of good, Ira, Darshini, good, Mahek. Correct answer. Ashish, correct. Yes, everybody put your answers in the chat box. This is a very simple one. Klinefelter's syndrome. Very good, Krishna. Correct answer. It is characterized by a karyotype of XXY. Right? See, Klinefelter's syndrome. How does it occur? Now, see, these disorders like Down syndrome, Klinefelter syndrome, and Turner syndrome, they are all occurring as a result of non-disjunction of chromosomes. Somewhere, this non-disjunction of the chromosome is happening. So Klinefelter's syndrome, it occurs by abnormal XX egg and a normal Y sperm. ठीक है, या तो क्या हो गया, एक egg है जिसके अंदर 2 X क्रोमोसोम प्रेजेंट है and एक sperm है which is having Y chromosome. इनका फ्यूजन हो गया है. All right, or a normal X chromosome or a normal X egg and abnormal XY sperm. So again, see, this is non-disjunction. This is non-disjunction. यहाँ पे they have not separated. This is non-disjunction. So in this case, the individual is going to have this genotype, right? Such persons, they are sterile males with undeveloped testes, mental retardation, sparse body hair, long limbs, with some female characteristics like gynecomastia. Okay, normally in case of males, we are going to find 1 copy of X chromosome and 1 copy of Y chromosome. But here what is happening? We are finding an additional copy of X chromosome. So थोड़े female like characteristics develop हो जाते हैं. Okay, and such persons, they are sterile males, they have undeveloped testes, rudimentary testes, mental retardation is there, gynecomastia, female like features, the body hairs are also sparse. Okay, so these all features are found in the Klinefelter's syndrome. Now, next, the abnormality occurs due to monosomy. The individual has 2n is equals to 45 chromosome with 44 plus XO genotype. Good, Mahek, Ira, correct answer. Correct, Krishna, Kashish, good, correct answer. Right, this is Turner's syndrome. This is Turner's syndrome. Turner's syndrome is due to monosomy. ठीक है, एक क्रोमोसोम कम हो गया. It occurs by the union of allosome free egg. X chromosome ही नहीं आया है in this case, and a normal X sperm or a normal egg and an allosome free sperm. Okay, this is going to result in the Turner syndrome. Again, Turner syndrome, we are going to find in females. Okay, because here there is absence of X chromosome. One X chromosome is absent. So again, we are going to find poorly developed ovaries. The uterus is going to be small, underdeveloped breast. All these conditions we are going to find in case of Turner syndrome. Good, everybody. Next, females with Turner syndrome have good. अभी पढ़ा है हम लोगों ने. Females with Turner syndrome have right. In the last slide only we saw that the females with the Turner syndrome, they're going to have small uterus, rudimentary ovaries, underdeveloped breast. So what will be the answer? The answer is going to be the all of these. Next question, all genes located on same chromosome. So this question is related to linkage now. All the genes located on same chromosome, what are they going to do? Yes, Darshini, Ira, correct answer. Mahek, correct answer. All right, suppose this is a chromosome and here there are various genes located. Okay, so all these genes, they are going to form one linkage group. Okay, so B will be the answer. All right, all the genes located on same chromosome, they form one linkage group. A linkage group can be defined as all the genes on a single chromosome that move as a unit during cell division. Okay, and these genes, because we are talking about linkage group, we are talking about linked genes, these genes, they do not separate until the crossing over, and if the distance between them is very less, they are not going to undergo crossing over. That is what is the idea of the linkage. If the distance between the two genes is very less, they might be linked. Okay, for examples, humans, they are going to have 23 linkage groups due to presence of 23 pairs of chromosomes. So please remember this point, very important. All right. Distance between the genes and the percentage of recombination shows. All right, Darshan, good. Ashish, good. See, the question is saying distance between the genes and percentage of recombination. Okay, what is the relation between them? Now you guys are getting confused between the relationship between the distance between the genes and the linkage. Now see, if I talk about the relationship of linkage with distance between the genes, that is an inverse relationship. Okay, why am I saying that it is an inverse relationship? Because for this case, if I am going to reduce the distance between the genes, if I am bringing the genes very close to one another, then the chances of the linkage are going to increase. Okay, that is why this is an inverse relationship. Now the question is asking about recombination. So see, the relationship of recombination with the distance between the gene is going to be a direct relationship. Distance between genes. How? See, if I am going to increase the distance between the genes, what is going to happen? The chances of the linkage are going to reduce, and the chances of the recombination, chances of crossing over are going to increase. Okay, so that is a direct relationship. If I'm going to reduce the distance between the genes, the chances of recombination is going to reduce because the chances of the linkage are going to increase. What will be the answer? A will be the answer. Question में recombination पूछा है, linkage नहीं पूछा है. Next question. If a genetic disease is transferred from a phenotypically normal but carrier female to only some of the male progeny, the disease is. Good. Darshini, Ira, very good. All right. Very good. Krishna, Mahek. Good. Right? What will be the answer? Sex-linked recessive is going to be the answer. Okay, sex-linked dominant हो नहीं सकता है because it is saying a carrier female. So if let's say there is some X disease, okay, so this female is going to be the carrier and this female marries a normal male. So in this case, what is going to happen? See here, what are we getting in this case? We are getting a male progeny which is diseased. Okay, so that is what the question is saying, that if a genetic disease is transferred from a phenotypically normal but carrier female to only some of the male progeny. Okay, to only some of the male progeny. So therefore the disease is sex-linked recessive. Moving on to the next question. Okay, see, the sex-linked recessive disease shows its transmission from unaffected carrier female to some of the male progeny. Example, hemophilia. The heterozygous female of hemophilia may transmit the disease to the sons. The possibility of the female becoming a hemophilic is extremely rare because mother of such female has to be at least carrier and the father should be hemophilic. Okay, so the answer is going to be D. Next question. Chalo, find out the answer. Good, good, everybody. Good, very good. Right? What will be the answer? Correct, absolutely correct, everybody. Very good. The answer is going to be GUG. Right? In case of hemophilia, on the sixth position of the global chain, what is happening? The glutamic acid gets replaced by valine. Okay. So the sickle cell anemia is caused by substitution of glutamic acid by valine at the sixth position of the beta globin chain of the hemoglobin molecule. All right. So what is happening here? GAG, that is getting substituted to GUG. Okay, and as a result of GUG, it's the valine amino acid which is being synthesized. Next, person having genotype IAID would show the blood group as AB. This is because, what is the reason for this? Good, very good, everybody, very good. Very good, Krishna, correct. Right? See the confusion, if a confusion can arise, it can arise between codominance and incomplete dominance. The answer to this question is going to be codominance. In case of incomplete dominance, what happens here? We are going to get an intermediate type of generation, which is different from both the parents, right? Recall karo the example of Mirabilis jalapa, flower color in Mirabilis jalapa. In case of incomplete dominance, the dominant factor is not able to express completely, right? The dominant factor is unable to express completely. That is what happens in case of incomplete dominance. But in case of codominance, what is happening? In case of codominance, both the alleles of a gene, they are expressing. Okay, so basically here we are going to find that both the characters are expressed equally. So that is what codominance is, and this is one of the classic examples of codominance. Next question. ZZZW type of sex determination is seen in. All right. Good, good, everybody. Ira, Kashish, Darshani. Good, correct answer. Everybody put your answers in the chat box. Yes, Krishna, put your answer in the chat box. See, in ZZZW type of sex determination, see this type of sex determination, it is found in insects, okay? In insects like butterflies, moth, okay? And vertebrates. In vertebrates like birds, fishes, reptiles. Here we are going to find this type of sex determination. Okay, so what your answer is going to be? Answer is going to be D, peacock. Now here in this type, ZW is female, okay, and ZZ is male. So quickly tell me in the chat box that this is an example of female heterogamity or male heterogamity. Quickly put your answers in the chat box. Here, which kind of heterogamity we are going to find? Female or male? Perfect. Very good. Very good. Kashish, Ira, Darshini. Very good. Right? Because ZW is female. So here we are going to find two different types of gametes. So this is an example of female heterogamity. Very good. All right. Next question. Which of the following is incorrect regarding ZW and ZZ type of sex determination? You have to find out the incorrect statement. अभी अभी हम लोगों ने solve किया. It's very easy. I told you in which organisms we are going to find this kind of sex determination. Very good, everybody. Very good. Everybody's answering correctly. Right? अभी अभी हम लोगों ने देखा था ZW. ZW is female and ZZ is male, right? So we are going to find female heterogamity. Right? We are going to find female heterogamity. So now here in the B statement, it is saying that the females are homogamitic and males are heterogamitic, which is wrong, right? And the question is asking us to find the incorrect statement. So B will be the answer. Rate of mutation is affected by good, Kashish, Darshini, good. Right? All of these is going to be the answer. Okay. Next question. Okay. Now, next question is the crossing of F1 to homozygous recessive parent is called. Very easy question this is. F1 को homozygous recessive parent से cross कराया है. What kind of cross are we talking about? Very good, Darshini, Mahek. Yes, everybody should be able to answer this question. Right? This is showing what? The test cross, right? F1 to homozygous recessive parent. If we take the example of height, what are we doing? Capital T, small t. This is the F1, right? If these are the parents, the F1 that we are going to get is capital T, small t. So this is crossed with the recessive parent. This is what? This is a test cross. Right? What are the genotypic and phenotypic ratios of monohybrid test cross? Quickly put your answers in the chat box. What are the ratios for test cross? Correct answer, Darshini. Very good. Very good, Mahek. Very good, very good. Right. 1:1. Chalo, next question. A test cross is used to determine what is the use of a test cross. Right? Good. What is the use of the test cross? It is used to determine unknown genotype. Right? Unknown genotype of a dominant phenotype. A will be the answer. Next, ABO blood group system is because of. Very good, Darshini. Correct answer. Kashish, correct answer. Ira, incorrect answer. Beta, read the options again. ABO blood group system. We're talking about ABO blood group system. Mahek, Krishna, correct answer. Good, good, everybody. Right? ABO blood group system. This is an example of multiple allelism. I gene and it has IA, IB, and I. These are the alleles. So multiple alleles are present. Right? So multiple alleles. Linkage reduces the frequency of. Read all the options carefully and then answer. Darshini, Krishna, correct answer. No, Kashish. Read the options carefully. Linkage reduces the frequency of. अगर linkage होता है तो recombination नहीं होता है, right? Recombination नहीं होगा तो फिर क्या होगा? सोचो सोचो, find the answer. Yes, linkage reduces the frequency of hybrids, right? Because linkage is doing what? It is reducing the frequency of recombination. Okay, hybrids. Next question. Distance between the genes and the percentage of recombination shows. Chalo, this is a repeat question. Everybody should be able to answer correctly. Good, Mahek, Darshini, good. Kashish, good. मेरा फिर से वही गलती अभी explain किया था बेटा. We have discussed the same question a few slides back. Good. Right, please read the question carefully again. What is it asking? It is not asking linkage, it is asking about recombination, distance between the genes and the recombination. They have a direct relationship. As I'm going to increase the distance between the genes, the recombination is going to increase. Next question, HbA and HbS alleles of normal and sickle-celled RBCs are. Very good, Kashish. Mahek, good. कृष्णा बेटा मल्टीपल एनालिज्म कैसे होगा? See guys, what is the question saying? HPA and HPS alleles of normal and sickle cell RBCs are. Now suppose that I have a person who's having HPA and HPS. ठीक है, एक normal है, एक mutated है. All right. So in this case, when we are going to check the RBCs of the person, we are going to find normal RBC also. Okay, and we are going to find sickle cell RBC also. Okay, this is basically a carrier of sickle cell anemia. Okay, we are going to find normal RBCs also. We are going to find sickle-shaped RBCs also. So basically in this case, what is happening? This is also getting expressed and this is also getting expressed. And that was the idea of codominance, right? Both the characters are getting expressed. Here, in the blood, if we are going to examine the blood of this genotype, we are going to find normal RBC also and sickle cell RBC also. So this is what? This is codominance. Carriers of sickle cell anemia is one of the examples of codominance. If I have this situation, both normal, I'm going to find normal RBCs. Right? But when I'm finding this condition here, I will find normal RBCs also, sickle cell RBCs also. So that means both of them are getting expressed. So this is an example of codominant alleles. All right, next question. If a colorblind man marries a woman who is homozygous for normal color vision, the probability of their son being colorblind is. All right. Good, Kashish, Mahek, good. I'll give you a hint. I'll give you all a hint. Color blindness. Acha, put the answer in the chat box. What kind of inheritance pattern is followed by color blindness? Yes, everybody. What kind of inheritance pattern is followed by color blindness? Autosomal recessive, autosomal dominant, X-linked, Y-linked. This is X-linked recessive disorder. Okay, it is X-linked recessive disorder. Now, what the question is saying? The question is saying there is a colorblind man. So, colorblind man marries a woman who is normal. So, he has married a normal female. Let's draw a Punnett square. XC, Y, X, X. What is the question asking? The question is asking the probability of their son being colorblind. Okay, so here, see, this is a carrier female, a normal male, again a normal male, and a carrier female, because male is responsible for giving the Y chromosome in this case, right? Whenever a son will be born, it's the Y chromosome which is coming from the male parent. Okay, so of course that makes the answer to be 0. The probability of their son being colorblind is 0. Good. Next question. A plant having genotype this is going to produce how many types of gametes? No, Kashish, incorrect answer. No, Darshini. क्या कर रहे हो तुम लोग? See here, it is a very simple question. There is a shortcut for such questions. Okay, if this is the genotype, all right, and the question is asking the kinds of the gametes. Okay, so the formula that you have to apply is 2 to the power of n in this case. All right, what is n? N is number of heterozygotes. Okay, n is number of heterozygotes. तो अब यहाँ पे देखो capital A, capital B, small b, capital C. How many heterozygous condition are you guys seeing in this genotype? Put the answer in the chat box. 1. Right? Ira, very good. ठीक है? तो क्या कर देंगे? We are going to place the value of n as 1 here, 2 to the power of 1. What will be your answer? ठीक है? Cross check करना है. देखो, ये वाला genotype दिया है. इसके अगर हम लोग गैमीट्स बनाएंगे वी जस्ट हैव टू पॉसिबिलिटीज बच्चा कैपिटल ए कैपिटल बी कैपिटल सी या फिर कैपिटल ए स्मॉल बी कैपिटल सी इसके अलावा और कोई पॉसिबिलिटी ही नहीं है so you are going to get 2 different kinds of gametes so what will be your answer is going to be C. Okay, I hope this question is clear to everyone. All right, perfect. Chalo, last question of today's session. A man with blood group A marries a woman with blood group B. What are all the possible blood groups of their offsprings? Chalo, very important question. Good, Kashish. Right, see what the question is saying. It is saying a man with blood group A marries a woman with blood group B. We have to find out all the possible blood groups of the offspring. ठीक है, कुछ भी नहीं दिया है question में. So what are we going to consider the genotype as? We are going to consider the genotype as IA and small I for the man, and for the women we are going to consider it as IB and I. Now let's make the Punnett square. IA and small I. IB and small I. Okay, so here IA and IB, IB and small I, IA and small I, and II. So now if we see what are the blood groups, for the first case the blood group is going to be AB, second is going to be B, third is going to be A, and the fourth is going to be O. So we are getting all the possibilities. So what will be the answer? A will be the answer. Okay,